Bacteria are ubiquitous across all natural environments. They are found in symbiosis with fungi, in the soil, in rivers, in glaciers, and they're even the reason why some sea creatures glow. Many of these environments are low on sources of food and shelter, making them harsh, competitive places to live. One harsh desert-like microbial environment is the Phyllosphere. The Phyllosphere is the above-ground portion of the plant. My research specifically seeks to understand the intraspecific competition of model leaf-dwelling plant pathogen Pseudomonas syringae. Intraspecific competitive interactions are when microbes of the same species compete with each other for food and other resources. These interactions are important to study because they drive evolution of their host and other community members. That dynamic can result in changes to their ecosystem as well. My focus is on how these microbes compete with each other using microbial weapons called S-type bacteriocins. Bacteriocins in general are toxic proteinaceous compounds that specifically target closely related bacteria. S-type bacteriocins are modular proteins that contain 3 domains: a receptor binding domain, a translocation domain, and a catalytic domain. They also encode for an immunity protein downstream to prevent self-killing. The sheer volume of S-type bacteriocins in Pseudomonas syringae genomes compared to other bacteria leads me to believe that these weapons have the potential to play an important role in their intraspecies dynamics. Encoding for a lot of S-type bacteriocins is costly, and they are even more costly to use. To deploy this weapon, the bacteria must act as a martyr and lyse to release the bacteriocin for the good of the colony. What I want to know is how we can use genomic S-type bacteriocin content to understand intraspecific Phyllosphere interactions, and ultimately how intraspecific Phyllosphere interactions change the population's genomic S-type bacteriocin content. I am doing this by mining bacteriocins from publicly available genomes and genomes of our own isolates, and I am in the process of performing inhibition tests in order to understand which strain kills which strain. When I am done with this, I will pin the bacteria against each other on plants and see if other factors in the Phyllosphere contribute to who ultimately wins. Why would I want to research S-type bacteriocins of Pseudomonas syringae? Well, first, agricultural plant health. Understanding interactions that increase or decrease plant health will give us insight into how to more efficiently feed a growing world. Second, preventing unintended consequences. Bacteriocins are looked at as potential biocontrol agents. Understanding their dynamics will help us understand what we may be doing to the microbiome when we deploy them in the future. Last but not least, biotechnology applications. Knowing these dynamics will help us know how to manipulate bacteriocins for future biotechnology adventures.